ऑडियो ना ये तेरा नहीं ना ठीक है.
Good afternoon, sir. I'm Dr. Halim Khan.
Yeah, very good afternoon.
Dear participants, good afternoon. Welcome back to the afternoon session. Today we have with us Professor Dr. Dilip Kumar Upriti, sir. He is acting director from CSIR NBRI, National Botanical Research of India, head Lichenology Laboratory, Plant Diversity Systemics and Herbarium Division. As I told you, he is from CSIR NBRI Lucknow. Sir is having research experience of more than 30 years in botany, especially lichenology, biosystematics, environment, climate change, bioprospection, and biodeterioration studies. To his academic qualifications, he is PhD from Lucknow University in 1983. Among his foreign visits, he has visited Germany as a Indian National Science Academy exchange program, received training on phytochemistry of lichenized fungi at Essen University, Essen, Germany. He had been to Antarctica, 11th Indian scientific expedition to Antarctica in 27th November 1991 to 1992. USA. He has visited also USA Field Museum Chicago during 13th to 17th May 2010 and delivered lecture on Indian Parameliaceae. To his credit, there is some awards and distinctions which I will read out. E.K. Janki Ammal National Award for Plant Taxonomy 2015, awarded by Ministry of Environment and Forest and Climate Change. He was awarded Fellow National Academy of Science India 2013. He was awarded for excellence, Award of Excellence by in 2013 by Organizing Committee of UGC sponsored National Conference on Resource Management and its Sustainable Use at LMS Government PG College Rishikesh. He was awarded by National Environmental Conservation Award 2013 by Department of Environmental Sciences, Bareilly College, Bareilly. He was also awarded Professor K.S. Ink Award 2012 by Association for Plant Taxonomy for outstanding contribution in the field of Indian lichenology. He was awarded the Razi Medal Award Taxonomical Studies on Indian Lichens. He was also awarded JBS Halden Memorial Scroll 1997 for distinguished contribution towards theoretical studies on environment by Forum on participated in leadership development program of CSIR during 2007. Participated in 11th Indian expedition to Antarctica, fellow of Society of Ethnobotanists. Was member of CSIR Steering Committee for Antarctic Research, awarded National Collaboration Fellowship to visit different universities. Worked as consultant India, Chandrabani, Dehradun, editor Phytotaxonomy, biodeterioration of newsletter, To consult pressure of society, botanist, pressure of environmental botanist India, proposed export in development Himalaya and its people by J.P. Pant Institute of Himalayan Environment and Development, Almora. Worked as co-director in 4th International Ethnological Conference, Lucknow, medicinal training course in 1994. Life memberships of scientific professionals. Unique achievements of discovered 104 species of lichen in science, more than 12 new combinations. Pioneer in India to study Antarctic lichens. Pioneer in India to study air pollution utilizing lichens. Pioneer in India lichen bioprospection. Credit has 27 PhD students awarded and 2 are to be submitted and 6 are registered. Number of publications are more than 350, 13 books. He handled more than 30 projects as principal investigator and co-principal investigator. With this few words, I would like to welcome Sir. Welcome, Sir, for presentation. Over to Pretty, Sir.
Thank you, Dr. Halim. But there is a different disturbance in your from your place, or your voice was not clear. Anyway.
Now it is okay, sir, because I was not known about the participants sound. Okay, sir. Now you can start, sir. Participants, you have made suspended the activities of participants. Over to sir. Sir, you can unmute yourself. Sir. Sir, your co-host. Now you can start.
Can you see the screen?
Yes, yes.
Okay, I will start now. This topic is about taxonomy and diversity of Indian lichens and their bioprospection. So before I start, I will tell you that it's a good afternoon to all of the participants and Dr. Halim. I'm Upreti from this place, from Lucknow National Botanical Research Institute. So whenever we talk about lichens, we know that this is a most neglected branch of botany, or particularly lichenology, and very few people they know about lichens. But in your Telangana or Hyderabad, this is very common. When you go for biryani, Hyderabadi biryani is famous. Its fragrance is famous only due to this lichen, and that species is Parmotrimma reticulatum, which is most frequently used. And in Hyderabadi biryani, they go, and it is that process called dum lagana. Dum of that parmelia or that lichen gives the fragrance to that biryani. If you see the past of lichen taxonomy in this country, it is the Britishers who started working on lichens, particularly the nature lovers of British Army. They started collecting lichens from different parts of our country, and they pass on that specimens to the lichen experts either in UK or Europe. So most of the lichens which are earlier collected or are worked out, they are not well preserved in our Indian herbaria. They are mostly at BM or somewhere else in European herbaria. But it was Dr. Avasti, my honorable teacher. He started this branch more systematically at Lucknow University, Lucknow, and he established a school of lichenology over there. I'm lucky enough that I'm his student there, and later on I joined NBRI. So, taxonomy of lichens. First, we know what is lichen. We all know that it is not a single plant. It is an organism consists of or comprised of two other organisms, one fungus and other alga. That alga may be a cyanobacteria, and they live together in a symbiotic association. You can find these both organism in nature as free state, means the alga which is most common, that is Trypoxia as a green alga, and cyanobacteria as a blue-green alga. You find them in nature. You find them, that fungus, in nature. But when they come and they establish or they create these lichens, a single unit, it is sometimes very difficult to identify them. And this combination of too many of these alga and fungi resulted about 20,000 species of lichens, which are known from this whole world. If we go to the classification, yes, it was Theo Foster, who is coined as father of botany. He terms this lichen first, and he considered that it is an outgrowth on a bark of trees, but it is called Linnaeus. He is the father of modern taxonomy. He was the father of modern taxonomy, we all know. He described those lichens as genus lichen, and he described about 80 species of lichens. But it was Eric Acarius. He was the person who has meticulously divided lichens into different genera, and it is they were published in a very well-known these books, Lichenographia Universalis, Synopsis Methodica Lichenum, in the 80s, 10 and 14. Then there was, they do not know about the dual nature. Then it was Simon Schroeder who demonstrated that lichens, they are not a single plant. It is lichen thallus is a composite structure. If you see that the taxonomy of these lichens is still in continuous flow, means up to the year 2050, yes, different lichenologists or researchers, they use different characters for segregation of taxa. Mostly it was based on the fruiting bodies, because it is the fungus which is more than 95% in lichens, and the reproduction is mostly played by lichens. So the classification of lichens is based on the fungi which is present over there and the fruiting bodies. So broadly we can say that we can divide lichens into pseudolichen, ascolichens, or lichen imperfecti. Then after the advent of these molecular tools in recent years, there is change in or flow in lichenology, taxonomy of the lichens. If we start with there are a group of lichens which are called primitive ones, means they appear as a dot like a structure on the surface. The fruiting bodies, they are not perfect, or they are poorly developed. So that group comes under Arthoniomycetes, or Arthoniales we call them. These type of lichens are very frequent in forest where there is more light, or more forests which are disturbed, or the bark of trees which is rough. So you will find this type of lichens, members of Arthoniales there. Then comes the initial stage of Lothidiomycete, which means the lichens which form a structure, a feature-like structure, perithecia, and mostly now this group comes, means it is the primitive stage of lichens when they form a feature-like structure. Then we divide them on that basis. Further, if you see, sometimes that feature becomes carbonized. It's become hard. Then it goes to that Eurotiomycetes, where these perithecia are there, but they are well organized, not in the earlier case. Then comes the calycials. Calycials means what happens when you know that the fruiting body apothecia is layered with a layer we call epithecium. That epithecium is, when not developed, this particular group goes to calycials, members of calycials. Then osteopagales, again, they are fruiting bodies. They are well placed within thallus. Then there is modification of this apothecia of the fruiting bodies too. Sometimes they stretch out and they appear like a worm or a lily, and that group called Graphidiales or Graphidaceae. We call them Arabic script lichens. Then perfect apothecia, they are there in Lacanorales. They are button-like structures. You can see very easily, and particularly this Lichanorales and its families within this family, within this order Lichanorales, most of the lichens of our country or throughout the country, they belong to this order. The other group means where the fungi in place Ascomycetes. If Basidiomycetes are involved, we call them Basidiomycota. And since this group is very confused with these mushrooms or Basidiomycetes, so only few species of this group are known. Again, this particular group requires a typical habitat, moist, rainy forest. So in our country, it is very few, means only few, 2 or 3 genera they are represented of Basidiomycetes in our country. The third group comes imperfect lichen fungi. Mostly the sterile lichens, they belongs to this group, and it is Deuteromycetes type of fungi. They are involved in lichenization of lichens from this fungi group. You can segregate lichens based on their substratum on which they are growing. They have an ability, means lichens have an ability that they can grow on any perennial substrate. So whether it is natural one or it is man-made artifact. So if they're growing on tree, we call them corticolous. Within trees, if they are more dominant on twigs, we call them ramicolous. If they are growing on stone or in rocks, we call them saxicolous. On dead wood, we call them lignicolous. Even lichens, they have an ability that they can grow on perennial leaves of most of the trees, evergreen trees, and ferns. They grow, and we call them folicolous, and they are considered as the most sensitive to the environmental pollution or in microclimatic changes. And on soil, we call them terricolous. So if they are growing on different substrates, we name them. For example, they grow on mosses, mossicolous. So on, we based on their growth or on which they are growing, we can classify them. Growth form of lichens, we all know about that. There are 3 major growth form of lichens. One is crustose, which is crust forming. That crust may be smooth, that crust may be rough, it may be cracked. So all they belongs to this crustose group. The foliose one, they appear a little bit leafy in structure. They are not tightly attached to the substratum. The margins are free, but the fruticose lichens, which are shrubby or pendulous, they are attached to the substratum from a single place, and they may be erect shrubby, they may be pendulous in their habitat. There are some intermediate growth forms too, means that crust may be sometimes is powdery, and we call them leprose lichens, or that crustose lichens, sometimes it is crustose in the center, but margin it is lobulate, or it forms some leafy structures. We call them placotoid. And sometimes these crust may be a raised one, thicker medulla, and it's sometimes imbricate or rising. We call them scammulose. There is again a group of lichens we call dimorphic. Dimorphic means 2 types of thallus are there. One is primitive thallus, primary thallus, and that primary thallus gives support to an erect structure which we call podacia, and that is secondary thallus. You must have heard about reindeer lichens. Most of these reindeer lichens, they come under this group, dimorphic lichens. When you go in nature, you must be confused with other fungi sometimes, or with algae. But how we can differentiate algae, bryophytes, and fungus? Fungus, they do not have that type of color which lichens have, because lichens they contain a small amount of algae inside them, so they appear as gray, not completely white as mostly the mushrooms or these fungus are. The bryophytes, they are more organized. They have more organized structures. Even in mosses, they have erect leafy structures too. And algae we know it's a different. When you see it grows in underwater, and they have different structure, shape, and size. What are the different, means the collection of lichens is a little bit different. as in case of higher plants, we use big sheets to press the material. But in case of lichens, if lichens are crustose and growing on a rock or on bark of a tree, sometimes we need a chisel and hammer to remove it along with the bark. If it is too hard, or if it's growing on a rock, then we use a geological hammer and chisels, iron chisels. There is we collect them in polythene bags in the field itself. Sometimes we try to segregate them in the field itself. But nowadays, it is must that you have to mention their global position system, or wherever they are growing, longitude and latitude. You have to mention, because such type of informations are useful for carrying out climate change studies in future. So when we collect them from rock, we use camera, or we can have a camera to take photographs. We use a 10x lens, because sometimes the lichens, they are quite tiny, or their structures or their fertile structures are not visible with our naked eye. So we use a 10x lens and a GPS system to locate, to properly write down its location. Similarly, as I told you, in higher group of plants, we have herbarium sheets, but the herbarium methods in lichens lichenology is different. We have these rectangular structure of packets, and inside we use a hard card sheet on which we put the lichen samples, or paste the lichen samples with some other light. For lichen identification, you can see that there are syringes, there is a blade. Earlier, the sectioning, because lichens, they do not have too many morphological characters. If you start from angiosperm and go down towards the viruses, the identification of the taxonomy of lower group of plants is more complicated. For identification of a single bacterium, you have to perform 100 experiments. Similarly, you will not find too many morphological characters for segregation in lichens. So what we do, we mostly depend on the chemistry. So within these syringes, we fill some chemical reagents. I will tell later on about them. And earlier we used to cut sections through microdome. It's a very tedious or long or time-taking method. Now we use a binocular. We use a this blade, and we directly cut the sections within that binocular. I will show you how we will cut that. So what are the different characters? When even in field itself, you can even segregate your material based on certain taxonomical characters. For example, on which substrate it is growing, what type of morphology, its color, or whether it has some reproductive structure. So you can differentiate them on the basis of their color, on the basis of substratum on which it is growing, and the fruiting bodies, whether it is apothecia, perithecia, or some vegetative structures. Even sometimes the attachment of organs, they are also used to segregate lichens in field itself, but you can see them only if you use a 10x microscope, the sand lens. As I told you that fruiting body is the main criteria in segregation of or in classification of these lichens. So that fruiting body may be an apothecia, or it may be a perithecia, or it may be a mesodymium. So apothecia, its shape, size, attachment, margin. So you can see down that it is the color. The color is different. Sometimes their attachment is different. Sometimes their margin is different. Sometimes they are lyrillate. So these are the characters which we use to segregate lichens. If that lichen containing a perithecia, so you can see the structure of perithecia. Sometimes these perithecia, they are naked. Sometimes they are half covered. Sometimes they are fully covered with thallus, or sometimes they bear some dot-like structures. We call them pseudosequelae, which are called aeration pores in lichens. Again, we go for cutting the sections of thallus. If you cut a section of thallus, you can see that only 5 or 10% of the thallus that contain green algae, where the vital activity of photosynthesis is going on. Sometimes that algal layer is restricted below the upper cortex. We call that when there is clear stratification of algal layer and medullary layer and upper cortex, lower cortex, we call them heteromerous type of lichens. But in case of Leptogeum or Colema, which are cyanolichens, they do not have that type of stratification. the nostoc beads are they are disturbed along the whole lichen thalli, inside lichen thalli. So we call them homoiomerous type of thallus. More than 145 species of algae, they get association or symbiotic association with three major types of fungi. And we know that it is the type of alga sometimes is very useful in segregation of genera. The major group, cyanolichens, which contains cyanobacteria, or chlorolichens, which contains green alga. So again, within these green alga, yeah, we know that is Shuboxia is the most common one. And again, in blue-green alga, it is the nostoc, which involves in lichenization. If we cut the section of these fruiting bodies, I have a vertical section of a peritheecium, a preacher-like structure. Again, we find many characters to segregate, means what type of thallus is there, or what type of this perithecia, whether the ostiole size is appearing, whether that involucrum or peridium is carbonized or not carbonized, its thickness, paraphysis, what type of paraphysis are there, they are branched, they are simple. So these all characters we involve in classification of or segregation of different species of lichens. Now I will tell you how we cut section. You can see that these button-like structures, the stage first method of cutting the section of apothecia, we use a blade, half broken blade. We see this apothecia through binocular and we zoom it maximum. So we cut a section when we means it's by practice that you can cut a section of 10 to 15 mm, means 10 to 15 upon 1000 millimeter if you view through this binocular. So you can see inside how this apothecia, when you cut a section, you can see there is epihymeneum, followed by hymeneum, subhymeneum, hypothecium, and there is a margin. If that margin contains algal cell, we call it laconorrhine. If it is devoid of that algal cell or restricted up to half, we call them urine type or biotin type, or we call them lecidine if there is no algal cells in the margin. Again, as I told you that in case of lower group of plants, there are few morphological characters. So the ascus, and particularly the ascus tip, it contains good amount of starch, but in a different pattern. If you put some iodine, sometimes that iodine reacts with the starch which is present in the thallus condition or the top of that Si, and it develops different structures. So these, based on those structures, sometimes the moon-like, sometimes the crescent shape, sometimes the broken in the middle, we established even genera can be segregated based on the reaction of iodine with the starch at the top of ascus, which is a structure of only 10 to 15 mm of size. You can you can see that how the arrangement of ascus tip or the structures at ascus tip, we use that character for segregation of taxa and type of spores. Paraphysis, as I told you, paraphysis, they are the pillars which supports the ascus. They are pillar-like in house. Similarly, these paraphysis, they support the ascus and they have different shapes and size in different genera or different taxa of lichens. They may be simple, they may be solanative, they may be constricted down, they may be brown at tip, they may be sometimes crystals all along, they may be anastomosingly branched. You can see spores. Spore may be simple in some cases. They may be transversely septate. They may be colored. They do not have color. They are sometimes longitudinally and transversely septated. They are sometimes hourglass-like. Sometimes they are pointed. Sometimes they have a round point. Sometimes they are brown. Sometimes they are red. So there are a lot of these characters of spores are used to segregate a species. Even their shapes of ascospores is used. As I told you that there is a crude method to identify the chemicals within lichens, that is spot test or color test we call. And we use this 10% aqueous solution K. We fill this solution in a syringe and we use we put that one drop on the top of thallus, then we see how the color changes. lichens, they have unique chemicals. And we know that these chemicals, you will not find them any other organism, most of these secondary metabolites. And when a particular lichen contains that chemical and we placed K on it, it gives some color. So with a crude method, we can say that, oh, this lichen is K plus and it has this type of dipside or dipside. Similarly, we use C. C is nothing. It's a bleaching powder which you use to be dissolved with bleaching powder or calcium hypochlorite. And we use it to know whether the lichen contains phenolic compounds or not. Because most of these phenolic compounds, they turn red when we put this C on it. Kc, it is similarly K followed by C. PD is this is paraffin diamine solution, which is 5% alcoholic solution we use. And again, it turns yellow when there is a dipside or dipside in the thallus. How we perform that? With the help of razor blade, we remove the cortex. As you can see that medulla will be visible, which is exposed or will be exposed. Then in a syringe, we put one drop of that K or C or PD, and you can see the changes in that color. So we predict, okay, if it is PD plus orange, then it may be fungal protosyntenic acid. If PD plus yellow and K plus yellow, it's salicylenic acid, which we confirm further either by microcrystallography, because most of these lichen compounds, they get crystallized into form and they form different structures. So there is a key is there, or there is some photographs are available regarding crystals of different lichens. And this is simple method. What we do, we take out a small part of lichen, we place it on a slide, and we put one drop of acetone from it. What happens after some times, this acetone, it dissolves the chemicals which are present in that lichen and form a ring-like structure on the covers in the slide. Then we have certain these reagents, particularly it is glycerin and acetic acid combinations. We put it and we heat it, then it produces these crystals. These crystals on their structure, on the basis of their structure, we can easily identify them. TLC thin layer chromatography again is a more refined method after confirmation of these color spots. So we use different solvent systems, and you all know about that how this TLC has done. And we first take out this lichen material into small test tubes. Then we put one drop of acetone, and with the help of capillary, we load those chemicals or dissolved lichen solution on this pre-coated silagel seed, which is available in market, and add up to different places. Then up to 13 centimeter, we draw a line, we dip it into different solvents. So lichen chemicals which has more molecular weight, they do not move more. But lighter molecular weight compounds, they move more. And we spray it with 10% H2SO4 and heat the plate up to 120 degrees centigrade, and it gives these spots. Again, there is a chart which was prepared by Culberson, a American chemist, lichenologist, and with the help of that chart, we can identify our lichens. We use some control. As you see that this first one is a control, which we know that it produces salicylenic acid and etranorine. Salicylenic acid comes at RF2 and the etranorine comes at 7. So we divide the whole 2 and 7 into different 5 parts, and we know the classes of what are the different class RF classes, relative flow classes, and we can even measure the relative value also. More sophisticated techniques are available, HPLC, GC, GCMS, LCMS, but they are more costly, and we only go when there is need of conformation of certain chemicals or a single biomolecule. We use those sophisticated techniques. We all know that after the advent of these molecular systematics or molecular techniques in in systematics, there is a tremendous change or in taxonomy of different plant groups, including lichens. Lichen, particularly, you must be heard about Parmelia. Parmelia, which was earlier known as a single genus, is splitted now into 30 genera. Yes, it is molecular techniques are there, or means whether we go for these ITS or nucleolus or rDNA or mitochondrial SSU. So that is again supported with some micromorphological characters. Alone we cannot do, we go for this molecular systematics or taxonomy that is not only based on these molecular tools, it is supported with some micromorphological characters too. If you see, you have seen that I have told about that these some lichens, how we segregate them. This is Cladonia. I talked about that. This is Reindeer Lichen, how we segregate them based on their cup structure. Whether this cup is proliferating from center or from margin, we segregate the species. Even in Asnia longissima, which is a very common lichen in temperate or alpine regions, is that you can see that those hanging old man beard, we called it. So the structure, for example, its attachment, the color of attachment, different type of vegetative structures. So based on those vegetative structures, it's a branching, whether it is filamentous, whether it is dichotomous, we segregate different species of Asnia. You can see that these are the structures, vegetative structures. Sections there is pigmentation inside medulla or outside medulla. Such characters are used. Now, if you see what are the lichenary sites in the country, we all know that it is Himalayas and Western Ghats. Himalaya, you can see luxuriance, but if you go for higher diversity of lichens, it is Northeast or Western Ghats. So they do not have luxuriance, but they have very high diversity of lichens. Again, we can divide the lichens of our country on the basis of altitude. Tropical area, we all know that it's about 0 to 1,500 meters altitude, and Sodia robusta is the most frequently growing tree in central Indian regions, or even if you go Odisha or that area. So this tree provided most suitable substratum for lichens to colonize, and we have recorded that more than 70 species of lichens they grow over there on these trees. Even a single, if you saw a Sodia robusta tree, what is there on the ground? Its roots are exposed. Then the lower trunk is laden with the soil. Sometimes it is a different niche. Sometimes a cracked and with soil. Different type of lichens they will come over there. If you move upwards, it is smoother. The branch are more smoother, and the twigs they have again a different pH. So the texture of the bark, the pH, and the water retaining capacity of the bark plays important in pollination of lichens. In temperate region, we all know that India, we have temperate region in Himalayas and higher altitudes at your south, where the altitude from 1,500 meters to 3,100 meters correct is under this area, temperate area. You have variety of trees, you have variety of subspecies there, so you have variety of lichens over there. You can see that even the most frequently colonized tree in temperate region, it is Quercus senicarpifolia, which we call black oak. Black oak is the most common is a tree which is most colonized by lichens. You can see large number of lichens growing on those trees. We have some areas too in Himalaya where we do not get precipitation or much water. Such areas are called cold deserts, and there you will not find trees, or tree line is restricted only up to 3,000 meters. In such areas, there are some exotic trees or some plantation of populus or salix trees are there. So we mostly the lichens, they grow on rocks, and those rocks are exposed to high UV. Therefore, they develop certain dark colored halide lichens. If we see what are the different centers in India which are responsible for lichenological researches, or which it is our National Botanical Research Institute, CSIR lab. We have more than 150,000 specimens with us, excellent infrastructure, excellent library, and if anybody who wants to pursue researches in lichens, he has to visit our institute. Then Agarkar Research Institute, they have rich collection of lichens from Western Ghats, and particularly they have good collection from Andaman Nicobar, so trematoid lichens. They have well-equipped laboratory for culture of lichens, Botanical Survey of India. You all know that it has 9 centers, and the centers mostly in Northeast and Calcutta and Allahabad, they are involved, or they have collections of lichens with them, and some researches are going over there, including Andaman Nicobar, one where Dr. Jagdish Ram is working on lichens. Swaminathan Research Foundation, they have good collection of lichens from Eastern Ghats, and they have developed a center of excellence in culture and molecular aspects of lichens. As I told you that it is the Britishers who started working lichens in India, and if you see all the research work which was carried out before 60s of the last century, it was compiled by my honorable teacher, and he has mentioned that up to 1965, the number of lichen species in our country was 900 only. But you can see the fast pace of researches which are going on, starting from 1960 to up to this period, that number has 3-folds more. Means now it is 2,700. Now in India, I think this number, I have a higher estimation of lichen diversity in this country, because a lot of regions, the biosphere reserves, a core zone of biosphere reserves, difficult terrains in Himalaya, they or even special habitats, they are not well worked out. So it's my estimate that this number may go up to 5,000 species from this country. Even you can see that there is very high endemism in our lichens of this country. We have more than 22% of lichens. They are considered as endemic to this place. As I told you that it is the Western Ghats and Eastern Himalaya which has highest lichen diversity in our country, but the central Indian region, which was frequently from the ancient time, it was under agriculture practices, so it does not have too much of forest, so it has very low diversity, lichen diversity in country. Himalayan region, higher altitudes of India, they are exhaustively explored for their lichens. And particularly the easily accessible areas, they are well worked out for their lichens. Some cursory collection of Indian lichens, most of the state of central region of India, Andaman Islands, because they cursory collections only they are there, but more systematic studies are now initiated by Dr. Jagdish Ram. Even most of the biosphere reserves, we have 19 biospheres in our country. They are not well worked out for their lichen flora. Alpine and difficult areas of India, still they are not well worked. So very quickly, I will show you there how much work has already been done by my honorable teacher or my senior colleagues or students who are working with us. So it's a joint effort that more than 64. Let us start with the past, that who were the Britishers who collected lichens. If you see that it is Peritote from 1840, Bellingner, Taylor, they collected these lichens from south. Then G. Watt, Taylor, they collected from east, and Stachy from Kumaon region or from Uttarakhand region. And they sent this material to either Islander or either A.L. Smith, who was a lady at UK, for their identification. And most of these specimens, they are preserved. They are not available with us. They are available in British Museum or other European area. Now you can see that after the Britishers, it was Professor Chopra, Kashyap, Chaudhary, and my honorable teacher Avasti. They started working on lichens collections, and we have, if most of the state of India, they were explored by CSIR, because we have too many projects to work on the floristic studies of different states. So we have more than 66 lichen taxa or genera, which are fairly well worked out from this country. But as I told you that the lichen taxonomy is still in a flux. So these taxa which were worked out long back, they all need revisionary studies. We have started revisionary studies on some group of plants also. Ms. Gravidaceae we started. Dr. Naika, he worked on Lacnora. Then Parmidot lichens, Dr. Divakar has revised them. Yellow lichens, Stylocystis, for Dr. Joshi, he revised. Some lignite CSE members or cyanolichens, they were worked out by Dr. Khare, yes, genus Asnia again. Then we have floras of some states also. We have flora of Himachal Pradesh. Yes, we have well worked out it. Jammu and Kashmir we have, Madhya Pradesh, Uttar Pradesh, Uttarakhand, Gujarat, Arunachal Pradesh, Assam, Mizoram, Manipur, Rajasthan, Sikkim. So we have some fairly good amount of information on floras of these states. Lichen flora of Kumaon Himalayas, for example, lichens of Uttar Pradesh. Yes, we have published. And even on monuments, we have worked out what type of lichens they grow on monuments and how they cause damage. Then terricolous lichens, then cyanolichens, or lichens which are growing on mangroves. So you can see that these are the publications. Even it's come on a book form. Then we have carried out environmental and air pollution monitoring studies, and there again we have studied the flora of that. Means without knowing the taxonomy, you cannot go for other aspects of lichenology. Either it's air pollution or it's bioprospection. So we tried that. We also worked out on the floras of different cities or some places of different states. If we go for conservation of lichens, no effort so far has been done for conservation of lichens. We all know that there are a large number of conserved areas or protected areas in our country. Within those protected areas, somehow lichens they are also protected. But we have argued that we should go for creation of lichen gardens, and we can declare some lichen sanctuaries, and based on the specific type of lichens growing in a particular area, we have mentioned about 25 lichen sanctuaries in our country. Similarly, our efforts to develop a lichen garden is here. The Forest Department of Uttarakhand, they have established a lichen garden. Probably it might be a first garden in its type in the world. So they have established a lichen garden in Uttarakhand. Now I will tell you what are the different aspects of lichenology or lichenological researches which can be carried out if you have a little knowledge of lichens on it. Started with forestry, means yes, if a forest is thin out, disturbed, you will have a different type of lichens growing over there. But if it is dense, if it is undisturbed, it has more moisture, different type of lichens will grow there. So by studying the type of lichens on that particular forest, we can predict the forest condition, its health, whether it is disturbed forest or it is not disturbed forest, with the help of lichens. Environmental pollution, we know that lichens, they are excellent bioindicators or bioaccumulators. Even in remote sensing or climate change studies, because lichens very quickly, they are considered as the best bioindicators or biomarkers, because they very quickly show the changes within their morphology, within their anatomy and physiology, if there is a change in environment. So for climate change studies, it's the best material. DNA barcode, we all know that. We can go over phytochemistry. As I told you that they have unique chemicals. You will not find such chemicals in other group of plants. Even the regulation retreat or such type of studies, they can be carried out. Even nobody has so far carried out what is the association of lichens with the invertebrates, or how the birds they use them, or how the reindeers they feed on them. Then again, we go for nanotechnology, because it's again a excellent area. If you see this figure, it is alga. We all know that vital activity of photosynthesis is going on in alga, and with that it produces glucose, erythritol, ribitol, sorbitol. What happens? Since this alga is penetrated by the fungus Ostoria, so fungus, depending on the type of fungus which is involved in lichenization, there may be three pathways: acetate polyvinate pathway, melavinic acid pathway, cyclamic acid pathway. It depends on the type of fungi which is involved in lichenization. So what happens that carbohydrate goes to the fungi, and fungi, depending on the type of this pathway, it forms or it manufactures the chemicals. As I told you that more than 1,300 secondary metabolites, which are unique in plants, in lichens, they are not produced by any other group of plants. there are certain chemicals which are common with other group of plants too. For example, anthraquinones, xanthones, nomones, steroids, carotenoids, they may be similar with other group of plants, but most of these chemicals which have come out from a straight polyvinate pathway, like dipsidizones, they all are polyketide or aldehyde group of chemicals. They are not produced by any living organism in this earth. If you see the status of medicinally important lichens in India vis-a-vis world, out of 20,000 information on only about 160 lichens are known from our country. That is based on ethnobotanical leads and other preliminary studies which we carried out. But 500 lichens information on medicinally important lichens, particularly from the folklore or from the ethnic use persons, it is there. And it is mentioned that there are 40 different diseases they can be cured with lichen compounds, an antiseptic, antifungal, antiasthmatic, cold, cough, or stomach against hydrophobia. Wherever you see, it is there. If you see these chemicals, they are unique. They have very functional sites, OH or CH or this one. So they are excellent material for development of nanomaterials, because such type of chemicals you will not find in other group of plants. So lichen compounds, they have very functional or active sites, easily used for development of nanomaterials, and we are working on that aspects too, because we do not find good biomass, or we cannot get good biomass of these plants for commercial exploitation. So we have to switch over to nanotechnology or nanomaterials. You can see that, and we have done that one. All we know that there is this Candida albicans. It is responsible for dermatitis or skin diseases, and it has 3 different growth forms. It might be mycelial, it might be virulent, it might be saccharomyceal. The ointment which is available in market is fluconazole. Fluconazole only can control when that Candida albicans is in mycelial form. So what we have done, we have developed nanomaterials from fluconazole, which is available in market. We have isolated one flavonoid from this Asnia longissima, because it was the leads was available that it's chemical asnic acid, or it has ability that it can earlier it was used as in different skin diseases. So what we have done, we have extracted one flavonoid, quercetin is common. We characterize it, and we have developed its nanoparticles together with fluconazole and a breakthrough in therapeutic mycology to develop a antimicrobial through combination of these enzymes. What happens? This quercetin initiated the production of furcinol from fluconazole, and the efficacy of that drug or that nanomaterial increases 250 times, and ability to control all 3 growth forms of Candida albicans. You can see that even the silver nanoparticles we have developed from this Asnia, and you can see the chemicals. Again, the chemicals, particularly which are from malevolent pathway, they are unique in lichens and malevolent pathway. In India, if you see that the exploitation of lichen is very common. From your place in Hyderabad, I know that this huge amount of these lichens, they are collected as spices or to give fragrance to biryani and other muttons, and they all are their trade secrets. They never tell, or it is not available in black and white, but it is a common practice in foothills of Himalayas. And you can see that trucks of loads of these lichens, they are exploited from Himalayas, and it is estimated that more than 10,000 tons of lichens, they are destroyed or exploited commercially from Himalayas. Again, lichens, you have heard about Kannauj. Kannauj is a place in Uttar Pradesh which is famous for its perfumery, and there is a perfume, we call it attar, which is prepared from lichens, and we have carried out studies that it is mostly the sparmidroid lichens, which are bigger in shape and size, and particularly within sparmidroid lichens, 3 species of Parmelia, Parmortima reticulatum, Parmortima niglensis, and Ostrocynensis, they are exploited more for development of that henna attar. It's a perfume. What they do? They just boil lichens. They take out that that fumes and pass it on sandal oil, and that is called henna. Again, we have a project where we have estimated the potential of lichens in dyeing, and you can see that there are 2-3 methods of dyeing of lichens. One method we call it is boiling metal, and one method is ammonia method, and third one is with some solvents we use. But this ammonia method is very time taking. It took a month to color a fiber, but this water boiling method is easy. And what happens when we put some lichens together with a fiber, particularly wool, silk, and we keep it for a week to cover it. So fermentation is started, and when there's a water, hydrolyzation is there. So lichen compounds, dipsides, and particularly that dipsides is broken into oxalic acid. That oxalic acid decarbonate because we cover it with cover that lichen and fiber. So the decarbonation resulted in orsinol. This orsinol, after condition of when nitrogen from ammonia reacts or present there in their solvent, that orsinol changes into orsin. This orsin is the compound which is responsible for giving different color based on the mordant which we use. Means mordant, what we use, how we can increase the pH. How we can increase the pH? If we can put some salt, if we can put vinegar on it. So most of the secondary metabolite of lichens have ortho-hydroxyl aldehyde group, as I told you, which reacts with the free amino group present in natural protein. What happens? Mostly the fiber, wool, it is of animal origin. If like pashmina or any wool we see, if it is of animal origin, it contains protein. So this free amino group present in natural protein forms a stable shift base, which ultimately impacts or imparts the color. That color may be changed when with the moderates. So this is all on the right side. All these lichens are there, and you can see on the left it is the color which they give. We all know about this Acroleachia. Still, this lichen is grow very high in our country at 4,000 meters, but still in Scandinavian countries where it grow very luxuriantly, still it is used for dyeing silk and wool, and particularly the tweed type of wool it is used. We all know about Rossella montagni, and it was earlier used as litmus dye. When you were in high school, you have used that litmus paper. It is the chemical dibenzofuran which was extracted from it, was used earlier. Now it is synthetically available. Not this lichen is there, but its dibenzofuran is the same which is synthetically produced. We under this project, I have this on right side, the Indian lichens, and you can see that this is the fiber which we have procured from Varanasi, because this Varanasi in our Uttar Pradesh is famous for producing silk. So we have used this silk from Varanasi, and the color which these lichens they produce, you can see this herbal color or natural color. You will not find such color in synthetic ones. But it is not true. Means we have to go for conservation of these plants. If we tell somebody that this lichens is used for this color, they will exploit it. So what we have done, we started culturing lichens in laboratory condition. Culture of lichens in laboratory condition is again difficult because you have to maintain two cultures, one for algae, another for fungi. But it is interesting that in lichens, it is the fungus which plays important role, and fungal apothecia or perithecia, these are the fruiting bodies which produce fungal spores. They can be used to develop mycelium. So what we do, we take out these fruiting bodies from lichens, we put that apothecium or fruiting bodies on the upper side of the cover slip. Then we put some this nutrient on the lower petri dish. So what happens due to this moisture, the ascospores they discharge. When ascospores discharge, we can see growth within 2-3 days. But sometimes some lichens are very, very difficult to culture even for their mycobiont. This is we call mycobiont culture, because for example, species of Osmia, they do not produce very quickly this mycelium. It took months together to get some biomass. But some species are there which are easily give mycelium growth or very quickly within weeks. Even we can go for broth culture also to get big biomass. And we are successful. In one case, we use this Asnia gardens. This is very common in Western Ghats. And you can see that in the center, it is the control one. You can see this white F1, F2, F3 is a wool which is not colored, or it's a control one. Then CDE, we used the natural thallus, and GHI here, it is the cultured mycelium. So in case of this particular lichen, this cultured mycelium is able to produce dye to the silk. Then we all know about lichens. They are the pioneer in colonization or in succession, and particularly a crustose lichen has an ability that it can grow on a rock, on a barren rock, where other plants do not. And they play important role in plant succession. Barren rocks, unstable for most of the plants, are excellent substrate for lichens. And what they do, they involve in geophysical breaking of the rocks. We all know that mostly lichen compounds, or mostly the calcium, magnesium type of substrates are available on the chemical, these metals are available in the substratum. And as I told you that lichens, they have very active sites. This OH or COH, which are polar groups, what they do, they catch hold of the calcium from the substratum, and what they do, they liberate this calcium into free state and assume its normal form again. So this type of practice is going on in nature thousands of years, and lichens, they are involved in soil formation. Again, what lichens they do, they adhere pressure on the substratum. When there is moisture, there is rainy water, lichens, they expand. When it is dry period, they curl and they break the upper surface or damage the upper surface. So such type of studies are useful for Archaeological Survey of India, who are the conservator of these monuments or buildings or old historical buildings, and we have to provide them the estimate of damage by lichens, whether a lichen is damaging their substrate or that building or not. So what we have concluded that it is particularly the crustose lichen, and if it is tightly attached to the substratum, producing secondary metabolite and penetrating the substratum, is more harmful than a foliose or fruticose lichens or a cyanolichen. Sometimes they do not produce chemicals, and sometimes they act as a cloth and they protect the monuments from very high temperature or other abiotic factors. So how these lichens, they can be used in forestry studies? Yes, as I have told you that forest condition, forest health can be predicted with the type of lichens growing over there. If a forest is untouched, is undisturbed, with more moisture, it will have different type of lichens. So based on these lichen communities, we call them bioindicator communities. We can tell you that whether that forest is disturbed or not. See this bioindicator communities. If this electroid, calcioid, or ostenoid type of lichens are growing in a particular forest, if they show their dominance in that forest, we can say that this is old growth forest or it is not disturbed one. If you have lichenoid or graphitoid type, yes, you can say that this is open, thinned out forest. It's a more wind or more light is there. That's why this type of lichens are growing over there. Lovarian and cyanolichen, mostly shady forest, you will have these Lovarian and cyanolichens. Xanthoparamidoid, peltitoid, they predict that this particular rock surface is not trampled by these grazing animals, or this is a stable rock substrate. Air pollution monitoring lichens, they are excellent indicators of pollution because they are very sensitive. They are true indicators, as I told you, that they very quickly show their changes within morphology, physiology, or in their chemistry. They are excellent accumulators. They accumulate different metals which are lethal for other group of plants beyond their physiological need. Even a single lichen, you can find that growing in the south, and same species you encounter it in Himalayas also, or lower foothills. So with the help of a single species, you can measure a particular type of pollution in a wide geographical area. They are perennial, they are slow growing, they have uniform morphology, they do not shed their organs. As the higher group of plants, they have leaf shed and everything. But in case of lichens, they show you cumulative accumulation of pollutant, which is 100 years or 1,000 years of old. They lack vexicuticle, so the metals which are there in environment, they directly come and fall out on the substratum, on the thallus of lichens, and they are easily absorbed or adsorbed inside the thallus. Lichens, they accumulate elements to concentration that vastly exceed their physiological need. As I told you that certain chemicals which are lethal for other group of plants, they accumulated in lichens. So in places where lichens are not there, we go for this transplant technique. In particular city centers, we go to by the nearby area of that city from where the lichens they are growing naturally, comparing a non-polluted area, we take out along with their substratum, paste those lichens on boards and hang those boards on poles at different altitudes and see accumulation of metals within 15 days or a week or a month. Because lichens, when they are live, they accumulate different type of metals. When they're dead, they accumulate different type of metals. Lichens an ability that they can accumulate trace elements, metals, metalloids, organic metals, inorganic metals, even radionuclides. And if we have past data with us, we can see individual lichen species, whether it is there or not, by revisiting. Then lichen zone mapping, it is an easy method or easy technique, or even a beginner can do that type of research work. We have done that type of research work in Lucknow city. We measured or we zoned whole Lucknow city with help of lichens, absence and presence. We all know that lichens, some species of lichens, they are very sensitive and they do not grow in city centers. So we have what we have observed that about a diameter of 9 kilometers, we do not find any lichens, but there is growth of lichens after that. So we divided the whole Lucknow city lichen desert, means there is no lichen growth that within 9 kilometers. It's the more polluted one. Then transitional zone, we have few growth of lichens, then foliose lichens. Since we cannot use, if machines are there, we know that we have machines which can measure the pollutant in a particular place, but we cannot fix that machine in too many places. But with this technique, we can measure the level of roughly the level of pollution in different zones of in and around the country. So lichen desert, we call it critical air pollution, inner struggle zone, higher air pollution, outer struggle zone, medium air pollution, transitional zone, low air pollution, normal zone, very low air pollution. It is mostly in the outskirts of the cities. If you have past data of lichen publications, even you can use that data, which we have used. That it was one Kurz who described about 53 species of lichens in 1860 from Calcutta city, and from that garden. You all know about that Calcutta garden. We revisited that area, even the same tree from where he has recorded that species or from that garden, and how the species combinations or how this community changes are there. We have predicted that it is urbanization which is responsible for the decline of lichen diversity. Even we have measured trace elements, metals, metalloids, and lichen is an excellent biomarker. We have suggested that whether it is genotypic changes or how this DNA degradation is there when we frequently spray with different concentration of chemicals for 45 days periodically, and there is a lot of changes in protein also. Such type of observation are useful, and it is particularly the lichens that pyxine cocos, which is very common in tropical regions, is an excellent biomarker to study. We have again studied, there is a lot of scope to study pH, how these polycyclic aromatic hydrocarbons, which are cancerous to other cancerous, can be studied or estimated with help of lichens. And we have carried out such studies in Himalaya during this voyage, during the pilgrimage to Badrinath. A large number of vehicles they fly over there, and how these lichens, these polycyclic aromatic hydrocarbons, which are carcinogenic, which are 16 such pH, are reported by US Pollution Control Board that these are carcinogenic, and how they are accumulated in lichens, whether they accumulated at ridges or at valleys, depending on the type of their molecular weights, means particularly high carbon, high ring or 6-ring pH, they accumulated in the valleys, but the light ring or 2 or 3-ring pH, they accumulated in ridges, lichens. So air pollution sensitivity of lichens to decose lichens, yes, we consider them most sensitive. Folios, they are moderate. Custos are least sensitive, and custos which are growing on rock or soil are considered as most tolerant. We have used lichens to date rocks also, and lichens which have a circular structure, we know their age, we know the age of certain lichens, particularly Rhizocarpon geographicum, which grows on exposed rocks after the glacier retreat. When glacier retreated, the rocks which are exposed, it is occupied by lichens, and lichens, they form circular structures. The age of this, the growth period of this lichen is 1 millimeter in 100 years. And with that particular yardstick, we have predicted that the Pindaric glacier was retreated 675 years in 1 kilometer in 675 years. How we do that? We know that we started 1 kilometer away from the glacier snout, and we measured the diameter of the Rhizocarpon geographicum on different rocks at 3 places after an interval of 50 meters, 50 meters. Then we have a correlation that the rock which is 1 kilometer away from that snout will have definitely a bigger size of that Rhizocarpon geographicum, because it was exposed earlier than a rock which is near the snout. So we calculated that this glacier retreated 1 kilometer in 675 years, which the studies are similar to the studies which are carried out by ISRO, Indian Space Research Mission. That's why they have involved us in their collaborative projects, and we are studying with them on climate change in Himalayan region in 5 states where they have established, I will tell you something about this also, how this climate change affects the plants. Climate change means rising temperature and low precipitation. When there is rising temperature and low precipitation, definitely species, they show shift, or they started climbing, or plants started to creep to higher elevations where they will get the same type of habitat. And what happens when there is high UV? High UV influences the morphology, or lichens, when they go for shift, or when they go for shift, we called it migration. For migration, there is a need of acclimatization. For acclimatization, we all know it is mRNAs which are responsible for phenotypic changes. Phenotypic changes means, yes, lichens, they start changing their color, means it influence how it influence the change in reproduction, or influence the cellular activities within a lichen. So this acclimatization leads to adaptation in thousands of years, means frequently or continuously, if they face such type of conditions for adaptations, then they go for these genotypic changes. Even certain cyanolichens, they have certain chemicals or amino acids. We call them microspin-like amino acids. They act as a screen, or to protect the lichens from high UV. And for the first time, we have discovered or mentioned these 12 Ma compounds in Indian lichens. You can see that these are all 12 these Ma, which we have estimated on different lichens in Himalaya. Again, the lichens, when they if we study their secondary metabolites, the secondary metabolite quantification increases when there is an increase in altitude, because when the altitude increases, more UV may be there, and lichen, to protect itself from high UV, it has to produce more of secondary metabolites within its thallus. So what is the best plant to see climate change? It is a species which is growing in a buffer zone, most sensitive considered with lichens which are growing on soil, or cyanolichen in a transitional zone, which will clearly show you shift. So we have to select such species. We all know that after 70 years or 80 years, the temperature will go rise 2 degrees centigrade and all. So to create such type of the temperature in alpine regions, we have, or with help of Wildlife Institute, Dehradun, we have set some these open top chambers in alpine regions of Himalaya. So within the this open top chamber and a control side outside, we can see a lot of changes within the vegetation, and within even if we go for estimating the chemical or physiological changes was the plant which are growing inside that open top chamber, we can predict that what will happen if the temperature goes 2 degrees increase after 70 years just at present. This is all about ISRO's project, which we have, where we are involved. And they have a multi-summit point, or what they have done, they have to see the shift. They have selected mountains of 3,000 meters, 3,500, and 4,000 meters, and at all four sides they have placed some sensors. These sensors are responsible for to know the precipitation, to know the temperature, and they are simulated with satellite. A person who is sitting at Ahmedabad can see what is going on in the changes in temperature and precipitation at 56 places throughout Himalaya for next 20 years, and how the vegetation is changing. We can use herbarium specimens too if they are well labeled or with perfect ecological notes earlier. As I told you that we have to mention about the GPS location of these specimens too. So herbarium specimen, they are excellent material for to study the climate change. Even we can see that how this carbon sequestration is there at that time on those specimens if we have past and present records of that. Thank you very much. I tried my best to finish it completely. Any query, any questions, you are welcome.
Thank you so much, sir. There are two questions from the participants.
Yeah.
How can we identify lichens? First question.
As I told you that starting from means it is there for the beginners. If they go in field, first they have to differentiate lichen from algae and bryophytes and fungi. Okay. As I told you, it is easy that a lichen appears greenish. It will not appear dark green. And so it is easier to identify first lichen, whether it is different from bryophytes or algae or other fungi. Then then some literature is available, some basic literature is available. We have two keys, means once you have an idea about that, oh, this is this particular genus, then how to reach up to that genus, again, it needs a little bit training. So if a person who really wants to, interested to pursue researches in lichens, he will definitely come to this place, NBRI. We frequently organize training courses. Even in your personal visit, with the permission of director, you can come over here. Dr. Sanjeeva Naika is here. A number of students are here. So basic knowledge or basic information, first you get, then only you can proceed further.
Sir, second question is it lichen means blue-green algae and chlorophyce with ASCO or Basidomycetes?
I told you that there are three type of fungi. That fungi, Ascomycetes, has a large number of different classes too, as I told you. Means sometimes if perithecia forming lichens are there, maybe the fungi which belongs to Dothidiales may be involved there. So different type of Ascomycetes fungi are involved with the different type of algae, different sets of algae. Means, as I told you, that there are 145 types of algae are known, species of algae are known, which involve in lichenization.
So, sir, means the combination, the participant want to ask, means the combinations except this blue-green algae and chlorophyce and Ascomycetes and Basidomycetes only will form.
No, lichens, but I will tell you, lichens which are growing along the marine belt or along the sea, sometimes they have lichenization with Phyophyceae members. Phyophyceae members may be there.
Okay. So, sir, this is my personal question. So, when they are growing on the bark of the tree, okay, when they are growing on the rock, I don't know, but when they are growing on the bark of the tree, is there any kind of response developed by the plant against these lichens?
No,
What
they go
attracts?
on. I
What
will tell you,
attracts?
they grow on dead bark. Okay, that dead part of that bark.
What attracts these colonists on the bark of the tree?
Yeah, I will tell you. For example, if a lichen spore is disturbed, is dispersed, it may be a fungal spore which is sexually produced. It may be a Isidia or Soridia, which is a vegetative structure. When they disturb from a tree, from a lichen, spore being lighter in its weight, it goes wide away. Then depending on the type of tree or substratum, if it is rough, it can be anchored over there. You will not find lichens growing in Taj Mahal because that is quite smooth surface. You will not find lichens growing on Taj Mahal. But next by, if you see one place where the construction material is cement plaster, which is rougher, uh, which can anchor the spores, lichens will grow over there. So depending on the type of bark, its humus is moisture retaining capacity. It it is that depends on the type of lichens to colonize over there. So lichens which produce by sexual spores, they have vast distribution. But Isidia and Soridia, being bigger in shape and size, will restrict to and show a only local or regional distribution.
Thank you, sir. One last question, sir. Is there
Yeah.
any host specificity for the fungi? I mean to say
For
lichens.
lichens, it
Yeah.
is there. It is there. Particularly, I was talking about feature forming or perithecia forming lichens. They prefer soft or thick bark trees. For example, smooth bark with more moisture. Particularly trees of Elnus here. And if in south, Dipterocarpus trees are there, they are preferred by one member of lichens. We call them thylotrimatoid lichens, and they are considered as indicator of rainforest, tropical rainforest.
One fine question. Are there any evolutionary theories how lichens are evolved or how lichens, any, I mean, it's progressive, what kind of.
Yeah, I will tell you. For example, if you see cyanobacteria is there, okay, means liganization, some cyanobacteria is there. It means what we consider the lichens which have the cyanobacteria, they may be considered as the primitive one, and they other species they evolved from there itself. It is the cyanolichens which are considered as primitive.
Thank you so much, sir. Thank you. Thank you
Thank you.
for. We'll be waiting again for your second lecture also. Thank you so much, sir.
On
For
23rd.
thank you,
Okay.
Prakash sir. Good afternoon, sir.
Good afternoon.
Prakash sir. Sir, you have joined in the meeting, Prakash sir. Sir. Good afternoon, sir. It is a time for us of our next speaker, uh, Professor Hs Prakasha, sir. He is UGC BSR Faculty Fellow, Department of Studies in Biotechnology, University of Mysore, Manas Gangotri, Mysore. His academic qualifications are MSc Botany and MSc in Botany with specialization Mycology and Plant Pathology, and MPhil in Seed Pathology, PhD in Applied Botany. His academic positions were during 1984 to 88 he was lecturer, 88 to 98 he was reader, 98 to 2018 he was professor. He was 6 terms chairman departmental studies in applied botany biotechnology applied botany and biotechnology in 6 terms. Teaching and research experience is 34 years. He guided 34 PhD students and 64 students for MPhil. His fellowships, awards, and other recognitions are UGC BSR Faculty Fellow, Fellowship, National Academy of Agricultural Sciences, New Delhi, Crop Protection, Professor V. Puri Award by Indian Botanical Society for the contribution to endophyte diversity, Professor S.N. Dasgupta Memorial Award by Indian Phytopathological Society 2014. He was Chairman Session Systematics of Viruses APS meeting San Diego, USA. Chairman Terminal Evaluation Team of FAO UNDP Government of India project on quarantine stations in India. Member Advisory Committee for UGC SAP DRCCS program at Bangalore University, Madras University, and North Maharashtra University, Jalgaon. IBSC of Coffee Board, CFTRI, DFRI, CFTRI Assessment Committees, LRC of DFRI, National Steering Committee, ICO, CFC Project on Coffee Leaf Rust on Coffee of Coffee Board, PRC 12th 5-year plan program of agriculture based labs of DRDO, Review Committee of 11th 5-year plan of DRDO DFRL, member of Vision Document Committee of University of Mysore, member of Animal Ethics Committee of CFTRI and University of Mysore, coordinator of research programs, Institution of Excellence GOI MHRD Award of rupees 100 crores to University of Mysore, coordinator of UGC SAP since 2004, DBT HRD program on MSc Biotechnology, DST Fifth Phase program, DANIDA program on training and research in seed health, principal investigator for projects funded by European Union, DANIDA, UGC, DST, DBT, ICAR, Advanta Private Limited. Research areas are molecular diversity, bioprospecting, diagnostics, seed health. He has national and international collaborations like Asian Seed Health Center with Danish assistance, developed collaborative programs with TSHC Denmark, ASHC Tanzania, Chinese Seed Health Center, Vietnam, Bangladesh, Nepal, European Union program, a collaborative research program on yield improvement of sorghum using innovative seed technology with Denmark, Burkina Faso, Tanzania. Visits abroad are USA, Denmark, Germany, Italy, Japan, China, Vietnam, Tanzania, Bangladesh, Nepal. To his credit, 200 research publications are there, out of which 127 are international publications and 71 are national publications. His citation index of publications is 2,915, H index 28, reviewed national and international research journals. He introduced new courses like MSc Biotechnology course, MPhil Technology at University of Mysore. He was Board of Studies Chairman in Applied Botany and Biotechnology, member of POS of various universities, editorial board. He is an editorial board of Journal of Phytopathology UK, Agriculture Research, published by National NAS, Asian Journal of Plant Science, Indian Journal of Biotechnology, He has organized number of national, international symposia, workshops, short-term courses with several collaborations. He attended more than 60 national and 20 international conferences. He edited 8 books, 7 bulletins, 6 leaflets on seed-borne diseases and mycotoxins. He has authored 15 book chapters. He has undergone various advanced trainings like JEOL Japan electron microscopy and DGISP Denmark seed health testing for viruses, IISc Bangalore molecular biology techniques, National Law School Bangalore IPR, ICRISAT Hyderabad plant pathology, CCMB Hyderabad electron microscopy in biotechnology, and Tamil Nadu Agriculture University Coimbatore. He had gone advanced training on biological control. With these few words, I would like to welcome Sir to deliver his talk. Over to Prakash, sir. Sir, Prakash, sir, please you unmute and make your camera on, sir.
Hello?
Yes, sir.
Can you hear?
Yes, sir.
Okay. Shall I proceed?
Yes, sir.
Okay, then fine. Yeah. Thank you, Dr. Khan, for your very elaborate introduction of whole of my biodata. What I thought is that today I will discuss mostly about the molecular taxonomy, but my emphasis would be on the barcoding. Can you hear? Is it clear?
Yes, yes, sir. You're audible.
The slides are okay?
Yes, sir.
Okay.
Screen is visible.
Yeah. So my main objective is to see the use of barcoding, especially the sequence-based taxonomy, especially for various groups of organisms. So I will try to give you an overall view of how this barcode, the DNA sequences, could be used for taxonomy purpose. So what is taxonomy? I think most of you are biology specialists, so I need not emphasize what is taxonomy, but to give you orientation, I will just define what exactly is taxonomy. So this is nothing but the biological classification. Mainly we group the organisms into different taxa. So we use different terminologies like classification, nomenclature, identification. For all these taxonomic terminologies, we use this molecular taxonomy nowadays. So most of the conventional taxonomy is being replaced with the molecular taxonomy completely, or at least it is trying to supplement the existing knowledge of taxonomy of various groups. So taxis is derived from the Greek word arrangement or order, and nomos refers to the distribute or govern. The groups that are making up the classification hierarchy are called the taxa. So taxonomy deals with classification, mainly the arrangement of organisms into different groups or taxa, mostly based on mutual similarity or also evolutionary relatedness. The nomenclature is mainly dealing with the assigning correct names to taxonomic groups, which is in agreement with already published data or information. The identification mostly refers to the determining that a particular isolate belongs to a recognized taxon. We also use another terminology called phylogeny. The phylogeny is mostly related to the evolutionary relationships between organisms. And however, the modern taxonomy, what we deal today, is mainly based on the phylogeny. So we do have a lineage for different groups and how these are related to one another and so on. So the molecular phylogenetics mainly deals with the nucleic acid sequences, either RNA in case of viruses sometimes, and also the DNA. And this is mostly based on the sequence which is used to establish evolutionary relationships between organisms. And we also use the terminology molecular systematics, which is nothing but the use of molecular data for taxonomic and biogeography purpose. We do have different taxonomic levels, and most of the organisms are grouped into domain, kingdom, phylum, class, order, family, genus, and species. The taxonomy is very, very important, especially in the biology field, mainly to enhance in order to know the fitness of the organisms to a given environment, how they survive in a given niche, and so on, and also to understand the inherent genetic variation among individuals and populations. And it has got very wide applications in evolutionary biology, conservation and management of natural populations. And also it helps us in organizing the huge amount of data or the knowledge about an organism. And also it helps us in predicting and framing the hypothesis for doing further research, which is based on knowledge of similar organisms. And also it helps us places the organisms into useful groups with precise name. That help the researchers to coordinate their work and communicate very efficiently. And it helps us in the validation and determination of the taxonomic resolutions of new identification tools also. The major approaches for the classification is one is the start with it started with Aristotle. He dealt with the artificial classification, although he could identify animals and plants. The natural system of classification, which was started with Linnaeus in the middle part of the 18th century, mostly which is based on the biological nature of organisms, although they had very scanty knowledge of microbes. So they didn't deal much with the microbes. The phylogenetic classification is mostly the post-Darwinian in 19th century or so, and they tried to look into the evolutionary affinities of various groups. And Ernst Haeckel, he separated unicellular organisms from plants and animals, and he created the Protista, which included the fungi, protozoa, bacteria, and algae. The modern system of classification started with the Whittaker, wherein he created the 5 kingdoms: prokaryota, including archaea and bacteria; protista, including protozoa and algae; fungi, plantae, and animalia. Of late, 1978 onwards, so for the first time, Carl Woese used some sequence-based information, especially the 16S rRNA sequences, and he created 3 domains of whole entire living organisms have been brought under 3 domains. One is bacteria, archaea, eukarya. This is mainly based on the ribosomal RNA sequences of microorganisms. So the first credit goes to the Aristotle as far as the taxonomy is concerned, and hello. Hello.
Sir, you lost your screen share, sir. Can you again repeat? You call that man.
I'll just find, yeah.
Down there is a green arrow. Up, share screen is mentioned there. Sir, you unmute yourself, sir. You're mute now. The first one, mic symbol is there. There you press it. You will get your sound. Please, you stay with sir for some time. No, please, I request you. Because it was good going. Suddenly what happened, I don't know. Please, you stay with him. Fantastic flow was there. Suddenly it misplaced. We were on Aristotle.
Yeah, got it. Okay.
Yeah.
Yeah. So I mentioned about Carl Linnaeus work. So he is said to be the father of taxonomy. He is a Swedish botanist. Mainly he used some morphological features. And also he has introduced the hierarchical structure of classification. And also the biggest contribution is his binomial nomenclature system. And the hierarchy what he has created also almost about 7 hierarchy he has created in the taxonomic groups. And Ernst Haeckel, German biologist, who is also a naturalist, philosopher, physician, and professor and marine biologist, and also an artist. So he discovered, described, and named thousands of new species. And also he mapped the genealogical tree relating all life forms and coined terms like anthropogeny, ecology, phylum, phylogeny, stem cell, and protista, and so on, which we are even today using these terminologies in biological systems. And he has also written a book, Tree of Life, in general morphology der Organismen. And I mentioned about the Whittaker's 5 kingdom classification, and he used different characteristics to group these organisms into 5 kingdoms. This was in 1920 to 1980 during those periods, and he worked on these 5 kingdom classification. And also the latest is the Carl Woese. His major contribution is the introduction of 3 domain systems in 1978, which is mostly based on the similarities in ribosomal RNA sequences. And this is the first paper published in Proceedings of National Academy of Sciences, USA. This is a paper of Carl Woese, which paved way for the introduction of 3 domain systems and in the classification systems. And this is the grouping of entire organisms, the living organisms, into 3 kingdoms. And one important thing what we have to know is that the oomycetes that were earlier being discussed under fungi have been grouped mostly based on these sequences, RNA sequences, into altogether a different group called heteroconta, that is tremenophiles, and which is closely resembling these algal systems. And this is also validated by the other characteristics. For instance, their cell wall structure, if you take the motile spores and so on, they closely resemble the algal systems than the fungal groups. And the prokaryotes show very high diversity in as far as the phylogeny is concerned, and metabolically they have different groups, metabolic characteristics. Then environment, they adapt to various different kinds of environments, and also the morphology also varies from shape of the cells to gram stain to the cell wall layers and so on. And pathogenicity also, some of them are pathogenic, some are not pathogenic, and so on. And the prokaryotic microbes, bacteria and archaea, they are said to be evolved along different lines, though both are essentially prokaryotic. The archaea is said to be the most primitive. They are common inhabitants of extreme habitats. And the archaea, they share some of the characteristics both with bacteria and eukarya. And we use polyphasic taxonomy conventionally to identify these organisms and group them under different species. And the species is said to be the basic unit of taxonomy. But as far as the microorganisms are concerned, bacteria, fungi, we also have some subspecies level classifications like strains, ecotypes, and these are said to be the lower distinctive taxonomic levels. And whenever we introduce a new taxon that needs phenotypic, genotypic, and phylogenetic information, and the classification of these microorganisms is largely aided by the studies on fossils and recently by the DNA sequencing. And the method of classification is constantly changing with the new technologies emerging, especially in the field of molecular biology. And most widely employed methods, they include morphological characteristics, different staining as far as the bacteria are concerned, biochemical testing, DNA fingerprinting or DNA base composition, PCR amplicons, DNA chips, DNA sequencing, and so on. In order to classify or identify different organisms, the very first thing is that we need to have reference stock culture collections. Mostly we identify a new organisms or identify an organism which is unknown, mostly with the help of the type cultures that have been deposited in various culture collection centers like ATCC and so on. So also we use typing methods. Sometimes if we use the biochemical profile for typing, then we call it as biotyping. Then the serological profiling is known as serotyping. If it is based on the phage sensitivity, then we call it as phage typing. Then the antibiotic susceptibility, and among all these, the DNA typing is the most preferred one, mainly because it is reproducible and it is easy to perform, and it gives a very high level of strain discrimination. And also, if it is based on the ribosome, the ribosomal RNA and so on, then we call it as ribotyping. And also, some time back, they were using this DNA base content, especially the G plus C content. And so they used to identify these organisms if it is within 3% for species and 10% discrimination for genera and so on. And the overall GC content ranges from 24 to 76% in bacteria. They also used DNA hybridization, which is an indirect measurement of the sequence similarity between the genomes. And a cutoff value of 70% similarity is considered for establishment of the species. And but however, this hybridization work should be used very cautiously, and it should be performed under highly standardized conditions. And why 16S RNA as a universal sequence for classification of microorganisms is because it is ubiquitous and functionally homologous. It is highly conserved, and it contains both variable and hypervariable regions, and it is extensively studied and represented in databases, and it is easily isolated and presented in thousands of copies in the genome. It can be easily analyzed, and also because we do have some very good databases, especially the ribosomal sequence databases for comparison, and functionally, this is similar across organisms. That means it mostly participates in the protein synthesis, and the sequences, they change very slowly, and generally, universal sets of primers are used to amplify about 1.5 kb regions of the genome. So this is a structure of 16S rRNA, and some hypervariable regions are being used, not the entire 16S rRNA regions. And the species concept by using these sequences is that so all the strains within a species must show similar phenotypes as far as the phenotypes are concerned. A designated type strain of a species constitutes the reference specimen for that particular species, and less than 97% similarity of the two 16S rDNA sequences that implies that it is altogether a different species, mainly for the ecological studies. But for actual taxonomic studies, if you want to identify a species or so on, then the discrimination level should be very low. It should be 98.5% cutoff value. When the similarity values are about 95%, then we may have to use other methods just to justify the creation of a new species. It also provides information on the differences between the new and existing genera, and these are subsequently aligned. Then phylogenetic trees are constructed, or dendrograms have to be constructed, and we do use different algorithms for treeing to prepare these phylogenetic trees, and mainly based on the maximum parsimony, maximum likelihood methods, and other techniques are available. The sequence data from curated databases are compared, and the multiple alignment programs such as Clustal X, Clustal W, and Clustal X2, etc., are being used mainly to compare the sequences, the homology of the sequences. And also it should include a type strain or type species if it is available. Then we do use different types of DNA markers, but I may not concentrate much on the hybridization methods and the PCR-based methods. But just to get a flow, I'll just quickly pass through these slides. So an ideal DNA marker should be highly polymorphic. It should be codominant in nature. The frequency of occurrence should be high in the genome, and it should be neutral, easily accessible, easy and amenable for easy and fast assay. It should be highly reproducible, and also the data exchange should be much simpler. So this is the RFPD. Where earlier days we used to use this method as one of the very first techniques that have been developed to compare the different species. Although this is not that reliable, the repeatability is low, and it is a dominant marker, so that it cannot be used to distinguish homozygous and heterozygous loci. And this is highly polymorphic. Then AFLP also was used for quite a long time. Typically 50 to 100 restriction fragments are amplified, and they are connected with the linkers, and they are sequenced, and so on. And this is again a dominant marker, and it is a quite cumbersome technique to say. Then we do have the markers which are still quite useful, especially in plant systems. Whenever in the breeding and all, when they are developing the markers for various genotypes, then they use these what are known as the variable number of tandem repeats, VNTRs in short. They do have non-coding regions, and they call microsatellite and mini or short random repeats, depending on the base space what we use for distinction of the species or the genetic groups. And microsatellites, a few to 100 tandem repeats are being used, and these are highly variable, and thousands of microsatellite loci occur in any given species. The greatest advantage is that these are highly variable, these are fast evolving, these are codominant, but the disadvantages is that it is quite expensive and time consuming. Then ISSR markers, that is inter-simple sequence repeats, and this mainly deals with a genome region that is between the microsatellite loci, and these sequences are also quite useful in plant breeding as markers of polymorphism. And we do have the star markers, sequence characterized amplified regions. Whenever we have some amplicons, distinct amplicons, then we can cut them and sequence them and use those sequences for identification of the organisms. Of late, we use barcoding. Barcoding is the mainstay now of late in the taxonomy of various groups. It is being extensively used across different groups of organisms, whether it is microbe or plant or animal and so on. The barcoding initiative started with the International Barcode of Life initiative. Almost about 25 nations have participated in that. Almost about 5 million species have been represented among 50,000 species and so on. And this is called the Consortium for the Barcode of Life, CBAL system. And they developed the global standards for identification of species. And also some European consortium for the Barcode of Life is also one of the active members of this group. And they created databases, especially the bold database, Barcode of Life database, INSDC, International Nucleotide Sequence Database Collaboration, and some of the labs like Canadian Center for DNA Barcoding and others are participating in this initiative. So the Barcode of Life database, the bold, is the mostly maintained by the University of Gulf, which is a public workbench for barcoding projects. And the researchers can assemble, test, and analyze their data using these workbenches and formulate as described various species of animals, plants, fungi, and other life forms. And also it provides life sequence statistics, the barcode clusters for animals and barcode sequences, almost about 21,000 lakh 45,000 around that barcode sequences are deposited. Then also international nucleotide sequence databases, mostly we use GenBank, EMBL, and DDBJ for homology searches and so on. And these are the public repository for barcode data records. Then I mentioned about the Canadian Center for DNA Barcoding, and there are several networks operating as far as the barcoding efforts are concerned. Then we do have, I mentioned about the GenBank, then the ribosomal database project, the RDP, Europe's collection of nucleic acid sequence data, EMBC, universal protein source, when we want to compare the protein sequences, the UniProt sequences, and also is being used. And whenever there is a non-availability of sequences, then the in vitro sequencing is being done. This is assisted by some eco-primer softwares. And also they use this compilation phylogeny by using some of the software tools. Then coming to fungi, fungi are said to be the highly cryptic. So the number of species may vary from 1.5 to 13.5 million. But among these huge number of anticipated species, only about 100,000 are being known and they are well described. So these are conventionally divided into 6 phyla, mostly based on the mode of reproduction, ultrastructural and biochemical characteristics, such as myxomycetes, oomycetes, and the true fungi classified in the chytrids. Then we do have zygomycetes, then the ascomycetes and basidomycetes. Also, we have another big pseudo division among these. These are called the deuteromycetes or fungi imperfecti, including molds and some anamorphic fungi. And this includes asexually reproducing fungi, some with well-known sexual phases in their life cycle. They are mostly phylogenetically classified among related sexual. Whenever you find the sexual stage, so they mostly belong to ascomycetes or basidomycetes. Then in mycology, we use terms like teleomorph, anamorph, and holomorph, mostly dealing with a stage of the life cycle. Teleomorph refers to the sexual stage, anamorph refers to the asexual stage, holomorph, the whole fungus, including anamorphs and teleomorphs, are called the holomorphs. The fungi that reproduce a teleomorph, they are placed under artificial phylum, the Deuteromycota. And but the concept is now almost obsolete, mainly because of the available molecular phylogeny tools, and which helps us in accurate placement of the species. Historically, the Article 59 of the International Code of Botanical Nomenclature has helped the mycologists to give anamorphs a separate name from teleomorph. But this practice is being discontinued as of 1st January 2013. The examples of anamorphs and teleomorphs I have just given: Pyracularia, which is asexual stage, the perfect stage is Megapotha grigia, and so on. The Amsterdam Declaration on Fungal Nomenclature, that is the 9th International Mycological Congress in 2010. So the International Committee on the Taxonomy of Fungi, so that has enacted a rule in Amsterdam that one fungus, one name. So we cannot use dual names for naming the fungi. It should be only one name. And some of the fungi have relatively complex and conspicuous morphologies. And many fungi have been detected using DNA sequences, but have never been seen. That is, mostly they are cryptic in nature. And such cryptic fungi are particularly suitable for DNA-based identification. In addition to species barcoding, the molecular methods are being used extensively for both phylogenetic reconstruction and species recognition. And many of the fungi reproduce asexually, making the application of a biological species concept very difficult. Moreover, fungal mitochondria are not maternally inherited, as in the case of animals. So it is quite difficult to use this cytochrome oxidase subunit 1 as one of the barcodes, which has been extensively used in animal systems. Slide man. So the environmental, so what we now use is the environmental sequence databases. For instance, Hibbett et al. And the classification and naming is based on the environmental DNA sequence, and which is not from the preserved specimens of the cultures. And they suggested for inclusion in the draft biocode, although they have not been cultured or seen or not deposited in the culture collections, just based on the environment sequence data. So it should be possible to group them, give some name, but these are commonly known as molecular operational taxonomic units, MOTU in short. So the minimum criteria required for naming a fungus, only based on the nucleic acid sequence, is that the genetic barcode standard must be included in the naming. At least 2 full sequences should be available, should be deposited. A published phylogenetic analysis demonstrating the monophily based on publicly available sequences, which are retrieved using BLAST or a similar method. The application of chimera checking software should be inbuilt, and the provision of locality data, including GPS coordinates, ecological substrate host data, etc., should be included. And it should be registered in a recognized public access deposits, online data repository, and so on, like Microbank. So Schwartz et al., he for the first time introduced the concept of barcoding for fungal identification. He used the nuclear ribosomal ITS sequences as universal DNA barcode, and he published this information in DNAs. And he compared 6 DNA regions as promising universal barcodes. The mitochondrial cytochrome oxidase and other coding regions have been excluded, mainly because these are being difficult in amplifying DNA and insufficient variability seen in these, especially in fungi. The nuclear rRNA ITS region exhibited the highest probability of correct identification of a wide number of fungal lineages. So that's why it is accepted to be a standard barcode for fungal systems, at least for a majority of the fungi. However, there are some fungi which cannot be easily identified by using these barcodes, the ITS barcodes. In such situations, we use the other barcodes that are available. So he tried various regions like 28S rRNA, which is said to be the long subunit, then 18S rRNA, small subunits, ITS, that is the post-transcriptional process, which is very much homologous to 16S RNA, which also includes a 5.8S region in that from 1 to 2. Then also a protein coding gene, RNA polymerase 2. And currently we have almost about 172,000 full-length fungal ITS sequences deposited in the GenBank, and 56% of them are associated with about 50,500 species and 2,500 genera. We use other barcode regions like translation elongation factor for fusarium, beta tubulin for penicillium, and so on. So wherever there are some difficulties in using this ITS, then we can also use some supplementary barcode sequences. So this is the region, ITS 1 to ITS 2. This region is being used for barcoding. And I mentioned about the use of 4 regions, the LSU, SSU, ITS, and RTB1. They tried across different groups, starting from the primitive group like Phylogenetics to Mesomycetozoa, and they looked into the PCR success. The PCR success for ITS region is said to be quite high across different groups. Okay, so then they also tried the barcode gap, which is called the probability of identification, and among that also they have seen that the ITS region, ITS, for instance, if you use the 4 barcodes, then the resolution is quite high. But even for just ITS alone, if you take, also is very good, very good discrimination you can get, even just by using single ITS region. So as good as a combination of barcode sequences across different groups of organisms. And also, if you take the normal distribution pattern and the ITS region, if you take across various groups, then it gives a very good histogram, which is very much desired, indicating that this could be a very good universal barcode sequence. Then coming to barcoding of plants, then I did not emphasize the problems associated with the barcoding of the flowering plants, and the existing taxonomic accords still need some modifications and updating and so on. And there is also a quite useful practical challenges of assigning unidentified specimens to known species, especially where the available material is suboptimal, like just juvenile or fragmented, processed, or where it is almost depleted, degraded, and so on. So in that case also, it should be possible to amplify these regions and sequence them. The plant barcoding mainly deals with plastic regions. Mostly these are the protein coding code barcodes like RBCL and MATK, and the non-coding spacer, TRNH and TSBA, and ITS region, combination of these. And it could be used in an entirety or just the ITS region. The studies focus on mixed templates and degraded RNAs, the just use of some environment samples, and they typically use this P6 loop, especially when it is fragmented in the plastic RNA region, mainly because of its short length and conserved primary sequences, and it make it particularly amenable to amplification and short-read sequences via NGS platforms. And it has got a lot of applications. DNA barcodes have provided a new biological tool for organisms, especially the biologists, to increase their understanding of the natural world. So over the last decade, 4 plant DNA barcodes, I mentioned RBCL, MATK, TRNH, PSB, and ITS2 have been developed, tested, and used to address some basic questions of systematics, ecology, evolution, and conservation, including community assembly, species interaction studies, taxonomic discoveries, and assessing priority areas for environmental protection. Also, it is being used in forensic investigation, especially when you're dealing with medicinal plants and medicinal sources and so on. So the DNA barcodes are being used in the for regulatory purposes also, mainly to monitor the commercial products such as foods and herbal supplements. And the major challenge is that mostly to build the global plant DNA barcode library and adopting this genomic sequence technology for efficient and cost-effective workflow in applying these genetic identification markers to additional fields of biological and commercial endeavors. So a major task for many plant systematists and field ecologists, evolutionary biologists, and others is the correct identification of a plant sample in a rapid, repeatable, and reliable fashion. And the DNA barcodes, which are mostly 400 to 800 base pair long, are being easily isolated. We do have them universal primers for amplifying them and so on. This was originally conceived by Hubbard et al. In 2003. And it combines the strength of molecular genetics, sequencing technology, and bioinformatics. And these DNA barcodes certainly offer a quick and accurate means to recognize previously unknown describe and name species and to retrieve information about them, and also has the potential to speed the discovery of thousands of plant species. So the beginning of the DNA barcoding for plants started a little late, although the concept has emerged in 2004, and it took shape only by 2011 or so, because it underwent a lot of changes, a lot of discussions, a lot of interactions, and so on to come out with a universal barcode. Certainly, we use some combination of these barcodes for identification of plants, and we can't say that it could be done with a single universal barcode. The primary use of DNA barcodes is for species identification, and we need not look into the entire life cycle of a plant. Even a small bit of any plant tissue should be okay for sequencing, and and also to quantify the consistency of species definitions across lineages of plants with a measure of genetic variability. The DNA barcodes are useful. So as a biodiversity discovery, also this barcoding is of use. And for the applied users of taxonomy, DNA barcoding serves as a means to identify regulated species, invasive species, endangered species, and so on. And DNA barcodes are now being used to address ecological, evolutionary, conservative issues, and dealing with the ecology, controlling of the assembly of species, and biodiversity fields, and so on. And also the interaction between plant and animal, okay, and so many other fields. So however, there are some certain inherent problems using these barcodes, and sometimes it may end up with the wrong identification, due to phenotypic plasticity and genotypic variability, and it may overlook some cryptic taxa and difficulty in providing reliable characters due to long maturity periods. This will provide the taxonomist conservationist a cost-effective, efficient tool, and it provides a faster identification service, especially for preserving the rare, endemic, and endangered species. But certainly, it is not as on today, is not an alternative to taxonomy, but useful supplement to support the identification of the plant system. And also, it is independent of age, physiological conditions, and environmental factors. It has the ability to detect the genetic variation beyond coding loci, and small quantity sufficient and efficiency is based on the detectable polymorphism. So it's universal, specific, and easy to employ, and the gene segment should be suitable for a wide range of taxa. It should have high variation between species, and it is routinely retrievable with a single primer set, and it's short enough to be PCR amplified, and unlike animals, we cannot use a cytochrome oxidase system for plants, and plant mitochondrial DNA has low substitution rate and rapidly changing and its gene content and structure. That is, it is quite unstable. So that's why we cannot use them for plant identification. And coding and non-coding sequences from the chloroplast DNA, along with the gene of nuclear DNA like ITS, are being used. And these are some of the gene candidates that one can use. But I mentioned about the PSB1A and TRNH1 here, and MATK, RBCL. And as far as the nuclear DNA is concerned, then we can use the ITS region. But just amplicons of ITS also they have tried, like Hypericum species and so on. The size of the ITS amplicon itself can be used to distinguish some species, but although we are not using this quite extensively. Then these are the barcode regions that have been tried to standardize the universal barcodes, and they have evaluated putative barcode loci across various genera representing various groups and so on. And of all this, if you look into this picture, then TRNH PSBA, if you see, it gives a very good amplification. The PCR success rate is quite high for this, and even RBCLA is also good, and even ITS1 is good for in some cases, not for all the groups, So, and also they used MATK. MATK alone may not be that useful. Okay. So, and also you can see the PCR success rate and discrimination ability and so on. The tRNH, PSBA plus RBCA, these two barcodes have given a very good amplification and resolution of species. And as far as the algal systems are concerned, mostly the classification is based on the type of thallus, whether it is unicellular, colonial, synovial, filamentous, siphonaceous, and thallus, and so on. And also they use the branching system, the attachment, manner of attachment to the substratum, type of chloroplast, presence of pyrenoids, and also other ultrastructural characters. So this is how they have grouped them, thallus, siphonaceous, filamentous, synovial, and so on. And they use the ITS barcodes, RBCL, and cytochrome oxidase, and also the translation unstable factor, TRPA, which is a protein elongation factor. And the sequence comparisons have been done, and it has altogether changed the whole conventional taxonomic approach, the grouping. For instance, even a colonial system with a single cellular system, or the branched thalli and so on, are grouped together. That means irrespective of the morphological features, if you just look into the barcode sequences, they are grouped together. That means evolutionarily, there might not be any distinction across the different morphological groups. So this is how they have been grouped now, and based on the independent evolution of unbranched filamentous type of thallus with within different lineages. Then coming to viruses, so I will not deal much about that, but certainly there is viruses are certainly easy to distinguish based on the sequence information, either the code protein or movement protein and so on. So they use the conventional system. They are highly varied, although look simple, but morphologically they are quite distinct. Different groups vary to a large extent as far as the morphology is also concerned. And there are DNA viruses, RNA viruses, and reverse transcribing DNA viruses, reverse transcribing RNA viruses like retroviruses and so on. And Baltimore has introduced the system of classification mostly based on the transcription and translation of the nucleic acid material, how the mRNA is synthesized from the native nucleic acid material. And even this system of classification is still valid today, even if you take the sequence information, So the number of species that have been identified is increasing every year, although it is quite old, but there are a number of species that have been, because there are almost about 5,450 viruses have been described. And also they have tried to introduce the concept of species into the viral classification system. There are also viroids, satellites, and prions, and so on. One example, if I can quote, is that, say, the identification of the type species, for instance, TMV and so on. Mostly we depend on the type specimens. So there is no type specimen as far as the viruses are concerned. Only the published information, the first published, authentic and accepted public information will be used as the reference source, for comparison. And they use the host system, the material in the trifolium reference and so on. And the common name they use. Take the tobacco mosaic virus, for instance, and this is well grouped, and we do have very well characterized virus. And this tobacco mosaic virus, they just italicize this common name and make it a species and so on. So they are trying to evolve this system of classification and mostly trying to universalize this system of classification, trying to develop the concept of species, genus, and so on. So that is still it is being going on, especially with the ICTV group. So use these sequences. So the viral nomenclature and taxonomy is still complicated till it is not complete, and so a lot of controversies are still there. And however, the number of taxa are being increased day by day, mainly because we get quickly the sequence information. As the sequence information is being generated, the number of species that are being identified also is being increased. Then, as far as the animals are concerned, so there is not much of dispute as far as the universal barcode is concerned, because across different groups of animals, the cytochrome oxidase arginate 1 has been accepted as the universal barcode, and it gives a very good discrimination across different species of animals. However, it is not suitable for barcodes for the reasons I mentioned earlier. And also for the birds, it's quite useful. They tried for birds also, again, the cytochrome oxidase. Even for the fish barcode initiative is also there. Still, they are using in that also the this barcode sequence. And also for butterflies, which is said to be one of the cryptic species complexes. Also, we can try this cytochrome oxidase barcode. So all said and done, so we should have a robust sequencing platforms. So we do have quite robust sequencing platforms from Roche to Illumina to ABI Solid and Pollinator and Anther and technologies and so on. So it is quite easy nowadays to get the sequence done, even for eukaryotic system and a big genome. All we can handle quite easily, and this is quite handy nowadays. And so this barcode concept may still further go on, and a lot of information may be generated and included in the taxonomic system. And starting from the first generation of sequencing, like chain terminating method of Sanger's, to the second sequencing, ligation by synthesis, including pyrosequencing, was being used quite frequently for uncultured or non-culturable ones. Then the optical detection systems with a fluorescent technology, which the Android uses. Then the grid ion system, which is said to be the very good technology emerging now, uses the nanopore exonuclear sequencing technology. And also the cost per genome is coming down drastically over a period of time, and mainly because of the improvement in the sequencing technology platforms and technologies. This is overall how all the organisms could be placed now, all living organisms in the web of life forms, as your title for the refresher course mentions. This is how it may look. And to conclude, so the cytochrome oxidase as a universal marker for species level identification has been accepted for animal groups. And the currently used barcode sequences for plants are RBCL and MATK and also tRNA-HPLC systems, plus the ITS sequences for plants. And ITS is a universal barcode system for fungi. And the biological barcoding system is likely to provide a new framework for cataloging, categorizing, and monitoring the biodiversity. And in few years, so the DNA barcode has moved from fantasy to reality. The markers, they differ in their ability to differentiate among individuals and detecting polymorphism in genome coverage and in the ease of application. They could complement one another depending on technical availability. And the molecular technology provides an independent approach for the characterization of life forms, and it will be more fruitful if an effort is made to integrate and the existing molecular fingerprinting data and coordinate the projects of molecular characterization. I think with this, I conclude my talk. So thank you.
Thank you very much, sir. It was very interesting talk on molecular level. Everywhere molecular biology has come, though the time has made a very great achievements in the field of molecular biology, and so now to get published our work, conventional work should be amalgamated with the molecular level things, and then only we are having a chance of getting published. So
Yes.
this will be a wonderful idea. I mean, you have told so many avenues of molecular ways of taxonomy. So especially, can I have my personal query that
Yeah.
how did this ITS was used in identification of fungi, especially fungi?
In fungi, this is accepted as a universal barcode because the databases are available for comparison. So in the GenBank databases, so this is being used for a large number of fungi. So ITS is a good universal barcoding marker.
And what will be the price of, I mean to say, making this phylogenetic tree of any species or any organism?
Uh, Maybe
Commercially,
about,
because
2 to 3,000 for, because this is a small region,
commercial.
Yeah, maybe about 3,000 or something like that, or just for ITS regions or
So
so.
participants, please can you put in chat box any question, because already our time is over. 4 o'clock it is. Thank you very much, sir, for a wonderful
Thank you.
talk and
Yeah,
motivating us.
yeah,
So thank you so much, sir.
yeah. Okay, then.
I hope this was four, and almost I don't see any questions from the participants.
Okay.
So I take this privilege on behalf of Department of Botany, Telangana University, and our director, Professor C. Manohar Azari sir, and participants. I would like to express my gratitude for accepting our invitation and giving such wonderful talk. So it was a very good journey for our participants to understand the molecular taxonomy. Thank you very much, sir.
I
Thank
also
you.
thank you for inviting me to share my whatever information I have. And also Professor Manorachari, this morning also he has called me and reminded me. And I thank all of you.
Thank you. With your permission, I would like to end the meeting now. Dear
Okay.
participants, please get back tomorrow also. And one more announcement is on Sunday also we have our lecture on both Sundays. This Sunday and coming up Sunday also we'll have the lecture refresher course will be continuing, because Academies virtual program should have no holidays. Offline programs may have one or the other holiday, but here we don't have any holiday. Kindly join us again back tomorrow at 9:45. Thank you one and all for joining with us. Thank you. Uh, I think Deshmukh madam has left, it seems. Okay. Somehow, thank you. Thank you very much, everybody. I will end the meeting now.